Welcome. My name is Ben Twining. I am the interim executive director here at the laboratory, and I am thrilled to see literally a standing room only crowd in our first event of the summer 2017 Café Sci series. I have no doubt that Barney Balch is the reason you have come out. He is a luminary amongst the local scientific crowd here. But again, welcome. My name is Ben Twining. I have been at the lab for almost 9 years. I came as a senior research scientist in 2008 and have taken on some administrative roles and have been the director here for the last 6 months. And I'm really glad to be in this role. So for those of you who happen to have just arrived and walked in off the street without any knowledge of what Bigelow is or what we do, we are a basic nonprofit research laboratory that studies the microorganisms and other things that live at the bottom of the food web in the ocean. So you're going to hear a lot about that tonight from Barney, but we study the microbes. We study the things you can't see with your naked eye, but these are incredibly important to the function of the ocean, the health of the ocean, and to our Earth. So they make much of the oxygen we breathe, they make much of the food that we get out of the ocean, and they take up a lot of the carbon dioxide that we emit from our daily activities. And so when you go home tonight, before you go to bed, you should thank a plankton, if you can find one. Bigelow is, as I said, a nonprofit institution. We get most of our money from the federal government to do research experiments and projects, and so we thank you, assuming you've been paying your taxes, for that support. It really is the bedrock of this institution. We get funding certainly from our educational programs and partners and from many of the services that we do for commercial entities. But we also get a lot of support, almost 20% of our budget, from philanthropy. And so I also thank all of you who have given generously to Bigelow in the past or might be considering it in the future. We encourage you to do that. In these times of tightening federal budgets, private support for research is more important than ever. And I hope you will agree with that. I also want to thank HM Payson, who is the sponsor of our summer 2017 Café Sci series. We're thrilled to have that support. I think this is the first time we've had a sponsor, and so we really thank HM Payson for making this possible. This is one of the things that federal research grants don't pay for, so we appreciate that support. I also want to say that I think that this is a time of critical importance for basic science research. I went into science, and in particular into oceanography, because I thought it was fun, I liked to be outside, I thought it was interesting, I liked to work on a team. But now that I have young children and I'm observing some of the changes we're seeing in our world, I think that the work that's done here and at other research organizations is more important than ever. And so I really appreciate the time you're taking out to hear about some of that research tonight. And I hope you'll come in the future and see some of our other talks. Towards that end, if you want to hear more about what Bigelow is doing, and I'm sure all of you do, I'd encourage you to sign up for our mailing list. I think there's a sign up when you came in, and feel free to jot your name down on the way out. We send information not too frequently, just the right amount, regarding talks that are happening here, exciting research results, all sorts of great stuff. So with that, I'm going to introduce Barney. Barney Balch is one of the senior statesmen of the Bigelow scientific community. Barney did his undergraduate at Cornell University. He's originally from Massachusetts. Did his graduate work at Scripps Institution of Oceanography out in San Diego. And then he went to the Rosenstiel School for Marine and Atmospheric Sciences, which some of you may know. It's a very well-known graduate school of the University of Miami, and was there for, I think, 8 years as a faculty member before coming to Bigelow in 1995. And that makes Barney one of our longest-serving SRSs, senior research scientists. But Barney's connection to Bigelow goes back even before that. Barney grew up in Rockport and got to know Charlie Yench, who was the founding director of the lab when he was in high school, and worked with Charlie, and may talk a bit about that, so I don't want to steal his thunder, but also worked up at Bigelow as a young student. And so Barney really is our connection to our founding, which is wonderful. Barney also is a fantastic jazz musician. You may know that. He plays the trombone. And he is the director, or I think the director of the Novel Jazz Septet, which is a wonderful group. And they'll be performing on Thursday at the Scadomfa Library. If you are not coming to our other event, you should come to that. And the other thing about Barney that I'll say before I turn the mic over to him is that Barney was the first person at Bigelow that I met. And certainly I can thank Barney for recruiting me to the lab. I met Barney on a boat in the middle of the equatorial Pacific Ocean, many, many, many miles away from land in December of 2004. And I was a postdoc looking for jobs. And Barney didn't offer me a job then. He made me take a job somewhere else before I came back here, or came to Maine. But that's when I met you, Barney, and I've enjoyed knowing you ever since then. So with no further ado, I got it. Before I hand it over, let me tell you a bit about the format tonight. So for those of you who are regulars at the Café Sci series, we're going to change the format around a little bit this year, and we're going to break it up into two parts. So Barney's going to talk for 15 or 20 minutes, and then we're going to stop. He's going to give you some questions to think about. We'll have some questions at that point. You'll be able to go to the bathroom, refresh your drinks, ask some questions, and then we'll start for a second section. And we'll see how this works. We hope that that will enhance the sort of discussion aspect of this rather than just the you sit and listen aspect. So with that, Barney, take it away.
Thank you. Can people hear me? No. No. All right. Can people hear me? It's on. Can you hear me now? Is that better? Do I have to yell, or is this voice okay? No. You can't hear me. More. Can you? Is that off? Yeah, we have a sound man here, so. Is that good? Okay. I want to make sure. Thank you for coming. It'd be a real drag if no one showed up. So today I'm going to be talking about the wavelengths of change, the impacts of yellowing in the Gulf of Maine. And to start, I need to introduce the group of people that make all this work possible that I'm going to talk about today. And some quick acknowledgments. David Drapeau, Bruce Bowler. These are senior research associates, Dr. Catherine Mitchell and Dr. Jason Hopkins, who are postdocs here. Very shy and demure Amy Wyeth, who is serving you wine out there. And Clara Bird, who is the research undergraduate that we have in the lab this summer. But the acknowledgments go on from there, and there are too many names here to go through everybody that has worked on what I'm going to tell you about. We run this time series that y'all explain in detail, and we've had a number of postdocs here, which are those it's supposed to be in orange. It looks more yellow. And I've had a number of undergraduates over the course of the years, and I've had a couple of high school students who have also helped integrally with what I'm going to present to you. I also point out Charlie Yench, as Ben said, the founder of the laboratory. He and Dave Finney started crossing the Gulf of Maine between Portland and Yarmouth, Nova Scotia, in the 1970s and 80s, along with a guy named Carl Boyd from Dalhousie. We incorporate some of their data into our database. And we've had a number of ships. One thing, when you work on ships of opportunity like ferries, they're commercial businesses. They come and they go. And so we've had a number of these over the years, but that have allowed us to maintain the sorts of measurements that I'll be describing. And funding of all this work has been by NASA. And you'll hopefully see the connection of why NASA would be interested in this as the talk progresses. So a little bit of history for you. Ben started telling you in the beginning. Charlie Yench, who was the director of the lab, the first director, the founding director of the lab, and after the talk, check out the little plaque on the wall behind the screen. There's a nice story about Charlie. He also established the field of biooptical remote sensing, which is using light and the properties of light reflectance and absorption to understand the amount of living phytoplankton that are in the water. And which is very important if you're, say, on the International Space Station, or if you're a satellite looking down at the Earth's ocean and you're trying to describe the color. So Charlie focused on the phytoplankton, the microscopic plants, and I'm just going to give you a very brief introduction to these. The ones I'm showing you here are Dionaflageolets. They have 2 little tails. They swim around in the water. They make harmful algal blooms that can discolor the water. Another group, the Diatoms. These live in glass houses, and I note the scale there. That's 1/500th of an inch that these cells are. The very biggest ones you can barely see with the naked eye. Everything else you can't see. But they come in these beautiful structures, and their glass houses are very elaborate. They're made of biogenic silica, which is a major mineral which sinks to the bottom of the sea. Another type of phytoplankton that is found out here, or at least was found in more abundance until about 10 years ago, these are called the coccolithophores. These are calcifying plants, and you could fit about 50 of, Emiliania huxleyi, you could fit about 50 of those in one diatom. So they're very small, and they're covered with these calcium carbonate, aka limestone, particulate inorganic carbon is another term, chalk is another term. These scales, they cover themselves with these scales. No one really knows exactly why. That's a whole other talk in itself, but they have these beautiful architectures. So those are 3 different very important types of phytoplankton that we find, and that, as Ben said, you should be very thankful to them. So what did Charlie Inch actually discover? Well, he discovered that the amount of plant biomass, the amount of material that is represented by them, is related to the reflectance of blue and green light. Fundamentally, it's a little more complicated than that, but that's the basis of it. So you start with a gradient of colors here that go from blue to green, and then you make it a little more scientific. You bring in the actual wavelengths of light. Green light is 550 nanometers, and this indigo, it's really indigo down here, would be 412 nanometers. And then you look at the relative amounts of that light being reflected from the ocean. More reflectance is in this direction to the left, and as you increase the amount of chlorophyll, that's the pigment that's in every leaf that you look out at the leaves out there, they're full of chlorophyll. As you increase the amount, basically there's more reflectance in the green and less in the blue. Okay, the chlorophyll absorbs in the blue and not in the green. And what Charlie discovered was the power of ocean color. And that is, this is a false color image of phytoplankton. This is mid-Atlantic bight all the way up to Nova Scotia. Here's the Gulf of Maine right here. And this is a false colored satellite image. And the red areas here are regions of high plant chlorophyll. And the blue areas down here are regions of low plant chlorophyll. Those are the biological deserts. And the green are areas in between. And even Henry Bigelow, who said in 1924, he was quoting the biblical epithet, "All flesh is grass," and he said, "All fish is diatoms." So those diatoms, that one group that I showed you in the very beginning, they are favorite food for fish larvae. And so think of the red areas here as areas that are full of diatoms. And these are the productive regions where there's lots of food for fish. And out here in this area called the Sargasso Sea, this is the Gulf Stream, by the way, which is meandering around. Out there's very little in the water, and the water is consequently very blue. So who cares? I can hear there's probably someone in here that says who cares. And the oxygen in every other breath you take is from marine phytoplankton. Half of the productivity on Earth, plant productivity, is in the ocean. Half of the CO2 produced by man as fossil fuel burning goes into the ocean, where some fraction of it gets scrubbed, gets taken up by these plants, and then they sink to the bottom of the sea. And that's called the biological pump. So the phytoplankton are taking up CO2 that we're producing, they're fixing it into their biomass, they get eaten by zooplankton or larger fish larvae, and they get pooped out, and that poop sinks right down to the bottom of the sea, where it gets sequestered for thousands of years. So if it weren't for the phytoplankton, the levels of CO2 in our atmosphere would be much higher than they are now. Phytoplankton are the bottom of the marine food web on which all life in the ocean depends. There are some exceptions to that. There are things like hydrothermal vents and such, but for the most part, I can say with good surety that the phytoplankton are at the bottom of the marine food web, and without them, the upper trophic levels, the higher levels, would have trouble surviving. So I'm going to take you now for a little short diversion. And first, is the climate around the Gulf of Maine changing? And this is a question I get all the time. How are things changing? And then there's another question. What is this about the yellowing Gulf of Maine? So we're going to deal with these two issues straight on. So I'm not going to show you a lot of data, but I have to show you a little bit of data. This is a science talk, after all. On this axis over here is years. And this goes back to 1890, this record. And this is the mean air temperature for 3 weather stations around Maine. One's in Acadia, one's in Corinna, and one's in Gardner. Why were they measuring temperature 100 years ago? Well, they actually were measuring temperature and precipitation because they needed to float logs down rivers. And so by knowing something about temperature and they'd know something about when ice out was, and knowing something about precipitation, they'd know how much the rivers are flowing. And there's a line drawn through there that's not drawn by eye, that's a statistical fit. And that line is highly significant. Even though there's a fair amount of variability around that line, there's about a less than 1 in 1,000 chance that that line, if you took a random sampling of data points, that you would achieve a slope like that. And what is the slope? The increase in temperature per year. The increase is 1.1 degrees Celsius per century. So it's 0.0114 degrees Celsius per year. So it's incremental, it's very small, but over the time scale of a century, you can see a very significant change. So that's the century scale trend over Maine for the mean air temperature. What about rainfall? Well, the easiest way to show what's happening to rainfall is to show the variability within a decade. So you take 10 years and you see rainfall smattering going up and down over 10 years, and then you come up with a statistic that you look at the variance, the variability within the decade. And this is in units of meters, and that variability has almost doubled over the last century. So that is to say that we have more floods and more droughts than we used to have way back in the early 1900s. So, and then the other little fact which is worthy of note is that 4 of the 8 wettest years in the century have been since 2005. 4 of the 8 wettest years in a century have been since 2005. So now I'm going to talk a little bit about tea. Any tea drinkers out there, you might appreciate this. Rivers carry what's called dissolved organic carbon. Think of it as colored tea. And it's really, it's the best way to describe this. You can have a hot brew tea, and here you see your hot water with a tea bag steeping in it. And then for you real tea snobs, they have cold brew teas. And cold brew teas are, as I say, they're extracted in cold water. I like the name of this one. It says it's called First Flush Supreme. And but you see the color of the tea is much paler than the hot steeped tea. Well, the same thing happens in watersheds around Maine. And I'm going to show you this with using a river record of this DOC, which is called dissolved organic carbon export, and that's in metric tons per day. And this is coming out of the second largest river that empties into the Gulf of Maine, the Penobscot River. And these are data of Tom Huntington at USGS, who lives just up River Road here. And he's been keeping track of the export of this tea out of the Penobscot River. And I show just a small part of his time series, showing you some of the wet years that we went through. And then the color of that line is the actual concentration. So the redder that line is, the more the hot brew that you're looking at. And if you look carefully here, all the fall discharge events are basically the hot steeped. And the cold brew are the spring freshets that happen after the winter. This one, here's another one. And so you have this variability that's happening through the watersheds as carbon is being washed down to the sea from soils and from organic matter that's in the soils. And it all goes, everything, including pollutants, it all winds up in the sea eventually. So how do we measure change in the Gulf of Maine? A number of years ago now, 19 years ago, we started this thing called the Gulf of Maine North Atlantic Time Series, or we call it NATS. And it's a transect that's roughly east and west that crosses 4 water masses. So we cross what's called the Western Maine Coastal Current, the Eastern Maine Coastal Current, Jordan Basin, and Scotian Shelf. These are all waters that have different physical properties, different temperatures, different salinities, and different optical properties. And we started this in 1998, and if you look out afterwards, if you're walking back to your car, you'll see a truck parked out on the far side of the building. It says NASA on the side. That's our laboratory away from laboratory. It's on the back of a flatbed truck. We drive it onto commercial ferries, and we connect up all of our sensors. We get water from the ship's sea chest, and we turn the vessel into an oceanographic vessel. So here are pictures from the MV Nova Star, which is the ferry before the one which is currently running. And here's our truck on the second car deck. And here, the circles here are pointing to places where we have radiometers, devices to measure downcoming light, upwelling light from the sea surface. Here is measured by a radiometer here. This is looking off the bow of the ship. Here we have a tub where we're incubating samples in water bottles that we bring back to the lab. Notice they're in there, the water is very clear. You can see the black caps of the bottles there. We'll come back to that later. Here's the gang working inside this 8x8x20 van. It's pretty tight. We have good music in there. And here is a satellite image, a sample satellite image. By using the ferry, we basically are very NASA-centric. We want to be out there during clear sky days when the satellite that's orbiting, and I haven't told you much about ocean color satellites yet, but when they can see the ocean. If it's cloudy or foggy, we don't want to be out there. I'm a fair weather sailor. I'm not proud. We always go out when it's crystal clear, or we try to. And this particular trip, it clouded up after we left Yarmouth, and those are clouds right there. But we got basically those little X's are 5 overpasses of satellites. Now these ocean color satellites, they're orbiting, they're polar orbiting. Every 90 minutes, they go around the Earth, and then beneath, the Earth is spinning, and they're going around the Earth like this, do this and chew gum at the same time, and they're basically mowing the lawn, and they're looking at swaths of the Earth, and every 2 days, they cover the entire Earth ocean. And so NASA can basically look at all the phytoplankton in the global ocean every 2 days. It's actually every 2.5 days. And there are a couple of different satellites that are aloft right now. One is called Modus Aqua, there's Modus Terra. There's one called MPP-VIRS, all different sorts of names. There's a new one that's going to be launched this fall, which is all very exciting. There's another one called PACE, which is still in the planning stages. So these are all follow-on missions. Satellites don't last forever. And so now we're using the HSV, high-speed vessel, Alakai, which also is called the CAT. And we have light radiometers here that are coming out of that little hole right there. We just got back yesterday from a trip, and it was a successful trip. We have, there's a viewing port there where we can aim optical instrumentation down into the water column. We also have 2 autonomous gliders. One is named Henry, and one is named Grampus, after Henry Bigelow's ship. And if you come to our open house, we'll have these gliders out on display. But these things go out for 24 days at a time, and they basically go along the transect. We put them in from a lobster boat here. They go along the transect over about this far, and then they come up to the surface every 6 hours. We talk to them. We say, okay, it's time to come home now, Henry. Henry turns around and spends another 13 days coming back. And then we pick them up with the same lobster boat 50 feet from where we dropped them. So there's a lot of neat new ocean technology here. The reason we have these gliders is we can look at subsurface distributions of phytoplankton. Satellites are really good for looking at the very surface distributions. The gliders are good for looking at the deeper distributions and giving us an idea all the way down to here is 200 meters. Our glider Grampus can go all the way down to the bottom of the Gulf. So I have a short little movie clip just to give you an idea. These gliders don't win any races. They go about half a knot. And here the diver's in the water thinking, oh, this thing's going pretty slowly. There's instrumentation, there's light sensors and so forth. And so the diver's just going to calmly turn around and catch up to this thing. And when he does, he sees that he's going to have to hightail it because that glider, even at a half a knot, is going faster than he would like. And so now he kind of loses it as he's trying to hoof it over back to the glider. And he's going to go underneath the glider. And you'll see some of the optical instruments that are putting out pulses of light, where we're looking at the fluorescence of chlorophyll. We're looking at the fluorescence of this dissolved organic matter. We're looking at optical scattering, and it is truly a marvel that these robots can go as far as they go for as long as they go. So now the diver's going to go underneath and watch right there, and you'll see some flashing lights here as they come into the field of view. There you can see those lights. And it basically has no propeller, and it basically changes its buoyancy, and it oscillates all the way across. It's highly efficient, and is why it can stay out there, and it only takes 220 Duracell batteries, copper tops. And so we had it programmed to go down and come up just once, and then it inflates a little whoopee cushion bag in the back to get its tail up in the air. And when its tail is in the air, it can make an Iridium satellite phone call home to say, hey, I'm here. And it uses its GPS sensor and so forth to locate it. It dead reckons. It does all sorts of cool stuff. Okay, so a bigger view. Here's the little Gulf of Maine here, but I wanted to show this. This is a satellite-derived estimate of dissolved organic carbon, showing now the St. Lawrence estuary, which is a huge volume of water which comes in, and also with the Gulf of Maine. And some of that St. Lawrence water comes along the coast of Nova Scotia and works its way into the Gulf. So we don't have just home-produced DOC here in the Gulf of Maine. We also have DOC which comes from this very large estuary up above. Here's the Nats transect right there. And we cross, that's the eastern Maine coastal current. You can actually see it in the imagery. So billions of kilograms per year, and here is the amount of yellow substance. It's technically called absorption, but just think of it as the amount of yellow material in the water. And there are a whole bunch of years here where we saw this actually pretty good relationship. And there's this one outlier here, which happens to be a year when we didn't have a ferry. And each of these data points represents many cruises across the Gulf of Maine between June and October, which is when the passenger ferries go. And this particular year, we only had one cruise because there was no ferry, and I had to pay for an expensive research vessel. So we don't know why that one is as low as it is, but these other data points, there are error bars buried in there. You can't even see them. The error bars are so small relative to the data points. But the bottom line is there is a relationship between the amount of DOC coming out of the rivers and what we're actually measuring, the river discharge that goes into the Gulf of Maine. So we're seeing some big decreases in the Gulf of Maine primary production. Remember what I said, that phytoplankton are the bottom of the marine food web on which all life in the ocean depends. And so if you stop the phytoplankton from growing, then the organisms that depend on them further up in the food web will have a bit harder time. And so here's what it looks like. These are data. We started measuring the growth of all the algae here in 2001 when we received funding to do it. And I want you to note, this is a factor, this is a logarithmic scale. It's like the Richter scale for earthquakes. It goes in, for every unit increase, it's a factor of 10. Well, there's a factor of 10 increase from here to here and from here to here. And you see we had the highest productivity we ever saw was in this 2006 time frame, and then we dropped to levels of about 30, something like that. Each of these are averages of many cruises across the Gulf, and then we had some massive drops here to about 20% of what they'd been. We had a little bump up here again in 2012 and 13. In 2014, we had yet another crash. And those error bars give you an idea of the range of variability. These are all phytoplankton. I show diatoms here, but all phytoplankton growth is represented by these numbers. The coccolithophores, they do one other really cool thing. They make these plates of calcium carbonate, of limestone, and they fix carbon, and they do what's called calcification. And if you look here at the scale, we cover 4 orders of magnitude, 1, 2, 3, 4 orders of magnitude. In the old days, we used to have very high calcification rates, and then they've dropped to the point in that low year, they were almost unmeasurable. The Gulf of Maine, when I moved here, the Gulf of Maine was a natural laboratory for coccolithophores. We'd have blooms that covered the entire, like 100,000 square kilometers. And we haven't seen any of those for a number of years. So there's some real questions about what's going on. So low years are 20% primary productivity. And calcification has become essentially unmeasurable. So I'm going to stop right here and we'll take some quick questions. I have some thought questions for you to think about while you're getting a beverage refill, if you'd like. Things like, why did the productivity decrease? What are the consequences of its decrease? What exactly is in this tea that I'm telling you about? It's not Salada or Earl Grey. Or Bigelow tea, sorry. How do we look at century-scale changes of color in the oceans? So we'll take a quick breather. You can stand up and ask quick questions, I suppose, or you want to go get a drink refill first.
Yeah, I think everyone has a quick break. Think about these questions in case you get asked.
There'll be a test
Come back in about 5 or 10 minutes. Keep going. Thank you. Okay, so Barney is going to continue the exciting saga of colored organic carbon in the Gulf of Maine. Barney, you want to see if there's any questions that people have been thinking about?
What was it? Is this on? What was it about 2010 that made all the numbers crash?
Yeah, so 2010 was just because of sampling. There was no ferry running that year. The previous CAT had stopped running, and so we rely, in the winter months when there is no ferry, we rely on research vessels, which are orders of magnitude 10 times more expensive, and we only had the money to do one cruise in the period when the ferries are usually running, and we might do 6 or 12 cruises.
But even the cocoa lifts dropped.
So indeed, the coccolithophores, that 2010 was, one of our hypotheses is that the river water which is coming in is bringing with it this T, which blocks the light penetration. It's absorbing in the blue where the plants need to have it, and it's blocking that. It's like putting an umbrella, so you're taking away the light from the phytoplankton, therefore they're not able to grow as fast. The other hypothesis, which was promoted by David Brooks, who is in this audience, a physical oceanographer, I can't see him, lucky him, he suggested that putting all that fresh water, 90 cubic kilometers of fresh water that got dumped into the coastal zone, would affect how nutrients from the North Atlantic actually are injected into the Gulf of Maine in the winter. So that's a nutrient-based hypothesis or a light-based hypothesis. It's a double whammy. You could have both, and with any plant, you deprive it of nutrients and light, they won't do well.
All right, we're gonna pass the mic around, and we'll do a couple questions, and then we're gonna have more time afterwards. So,
I was sort of a little bit confused by the last graph you had up there, and I was wondering what you see the trends are. I mean, I kind of reading the paper expected everything to be going down or everything to be going up, and yet it was pretty lumpy. It bumped around there, and so what do you see as the most recent trends from the data that you see there, and why is it so lumpy?
can you, if I turn mine on?
Hear me? Can you hear me? Yeah, okay. So there were changes. There was, in terms of rainfall, there were changes, decreases in rainfall. 2012 actually was one of the warmest years that the Gulf of Maine has had, so that's a complicating factor. And as far as the decreases in rainfall, we're still up in the air as to whether it's this dissolved organic carbon, which is blasting down rivers, versus the inflow. But you're absolutely right. It's bumping around. It's not fortunately for us, it hasn't completely crashed. But things like that coccolithophore production, which is now almost unmeasurable. We're processing the samples from yesterday's trip overnight as we speak. And you know, it's a complicated, it's complicated out there. And I'm not going to pretend to say that I have the absolute theory on what's driving it, except it is, you know, if you're a small larva that needs to feed on the phytoplankton, if there aren't any, that larva won't survive. So, you know, it has food chain ramifications. Oh, sorry. Turn it off.
We can only do one mic at a time, nonprofit. But it's a great example of how important time series are, right? If you only measured 1 year, you might get the wrong impression, so you need to measure all of these. All right, one more here.
Does the problem exist because of the lumber mills way up north, and the water coming from them years ago was brown, and I just wondered if there's any relationship between the amount of water now that we have the rain and the decay perhaps of the work, the scrub that comes from the lumbering industry has anything to do with this?
Yeah, so that's an excellent question, because there's also dam removal that is happening. There have been a number of major dams, and those are now allowing a particle load to come down the rivers that was not there before, because sediments gather in the upper part of above the dams, and now as they pull them out, all of those particles, which attenuate light as well, are coming into the Gulf of Maine. That's something that we have not factored in the analysis, mainly because I'm not an economist, a land use person, but the changes in land use have been remarkable from the time that Maine was clear cut down to stubs, and now with subsequent regrowth and then more clear cutting and such. So you're absolutely right that that's a factor that should be put into the mix. Unfortunately, I'm not an economist and deal with that kind of stuff, but the dam removal, I expect, plays an important part.
I think we should keep going.
Okay.
Well, good chunk of time for questions at the end. So
Okay. Okay. Well, now on this little journey of color in the ocean, remember I asked you about how do we look at century scale change in the ocean. Well, I'd like you to meet Francois Alphonse Forel, a Frenchman. He was considered the founder of limnology, the study of lakes, and his compatriot, Willy Ulli, a German limnologist, from 1892 and 1890. And Forel did it first. He composed a handheld color scale, a color comparator scale, with tints varying from indigo blue to cola brown. And he used this as a way to quantify the colors of lakes, streams, not streams so much because you could see the bottom of the stream, but lakes, coastal waters, and oceans. And the Forel-Ulli scale, or I call it the FU scale, has been calibrated to modern radiometric measurements by a guy named Fernand and Van der Voorde in 2010. And basically, in a nutshell, what they did, a color scale relies on the rods and cones and your color perception in your eyes. And we all have slightly different color perceptions. Some may be colorblind, some not. But in general, there's an average human perception of color, which in 1936, the French discovered, and they came up with a chromaticity scale, which I'll talk about a little later. Whereas radiometry, which is hanging an instrument called a radiometer that measures photons coming from the ocean, those are calibrated absolutely. They are scientific instruments. You could say your eyeball's a scientific instrument of sorts, but somehow we needed to make these two measurements match. We make radiometric measurements from the bow of the ferry. Forel and compatriots made chromaticity measurements with their eyeball, and they looked at this color scale. And the Forel-Ulli color scale looks like this, and it has tints from blue to cola brown. And you basically look at the water through it, and you compare the color to what the most similar color that you see. And that actually has really useful information about the optical properties of the water. There's a comparatively large data set of older FU measurements. And Henry Bigelow, the laboratory's namesake, made such measurements in the Gulf of Maine in 1912 and 1913. And we can compare Bigelow's colorimetric measurements to the NAT's radiometric measurements with appropriate conversions. And I have to say, I came upon this quite by accident. This is a copy, this is one single paper by Henry Bigelow. And this is half of it. Actually, the other half is in my office. I found this in an antique bookstore. And this sat on my shelf for a while. I never realized that Henry Bigelow made any color measurements. And then one day I was looking through it, and I saw this chapter called Color. And I thought, oh, that's kind of interesting. So let's look at what I found in there. Whoa, what did I do? There we go. So here's the Forel color scale for those of you who are sitting further back. And this is a copy of the original. And if you know your French, it says down below, 30% de solution jaune dans le bleu. So the percentage of yellow in the blue. And I want you to also note here along the bottom are the numbers, the percentages that go from 0, 2, 5, 9, 14%, 20, 27, 35, 44, 54, and 65. And you say, what's the significance of that? Well, you'll see in just a second. Remember those numbers if you can. So here is, this is Marcel Fernand, who is making Forel-Uli color measurements. Here's the full scale down here, showing the low numbers, 1 to 11, and then 12 to 22. So this is the Uli part. And if you really want to do this yourself, there now is an iPhone app to do the Forel-Uli scale from your smartphone. And so write this down, eyeonthewater.org. You can go there and download this app. It's free. And you can go to any water body you want, the lake out in front of your house, the Damascotta River, and you can download this thing, and you can make your own FU scale measurements. So here's Henry Bryant Bigelow and his schooner Grampus over here in 1914. Here he is at the helm, and he is our window to the past ocean color of the Gulf of Maine. So here's his picture again, and this is a page from that. Here's the chapter called Color, and I know you can't read that from back there, so I'm going to blow it up just a little bit, and you may still not be able to read that, but I'm going to read it to you. At the other extreme, we have invariably found the percentage of yellow greatest, 27 to 35%, in the coastal belt along the shore of Maine, out roughly to the 100-meter contour, with secondary smaller but very green areas, 27% yellow along the outer side of Cape Cod. And I thought, those numbers sound a little bit familiar. And down at the bottom of the page, he then gives, as a footnote, the colors, the percent yellow, 0, 2, 5, 9, 14, 20, 27, 35. And you probably, if you were in the town of Boothbay Harbor, could have heard my whoop down in my office when I saw this, because this was, in fact, this meant he was using the Forel color scale, and I had the window that I needed to go back and look at the Gulf of Maine 100 years ago. So, and then he has another page in there. He's got all the original data in percentage of yellow. And so I had Erica Eames, the high school student. She came in after school and she converted all those. She basically digitized all those, put them into the actual Forel color scale, and we put it into modern day computer software so that we could look now at the Forel scale as Henry Bigelow saw it in 1912 and 1913. And the color scale is shown here on the right. And basically, the redder the data are, the more yellow the water is. Sorry, I suppose I could have gone from blue to yellow to make that obvious. But basically, the higher Forel numbers here are yellower, and these light blue numbers here are lower Forel scale numbers. And none of these go into the Uli scale yet. They're all within the 1 to 11 of Forel. So I thought that's pretty cool. And you see near the coast, there is these higher numbers here, which means the water is yellower. And out in the middle of the Gulf, it's bluer. So we decided to take these data here that are roughly along the transect between Portland, Maine, and Yarmouth, Nova Scotia, and look at those specifically. And there's the data right there. Here's your Forel-Uli color scale. And this is longitude along the bottom axis. It goes from 70 degrees longitude, here 70 to 66 longitude off of Yarmouth. And basically, as you go towards the coast of Maine, the Forel scale the water gets yellower. That was in 1912. And the mean value for Forel that Henry Bigelow saw for the Gulf of Maine was 6.6, and that was the range. So I'm sorry about this slide in advance, but there's no other way for me to tell you how this works except to show it. So I'm going to try to make this as simple as possible. Chromaticity, as you'll remember, is related to the sensitivity of your 3 color-sensitive cones that you have in your eyeball. There are red sensitive, blue sensitive, and green sensitive. And back in 1936, they came up with this way of denoting this in an XY plane. Don't worry about it. but essentially, this line here is how your eyeball works with the relative sensitivity of your blue, green, and red color receptors. And what this guy Fernand did was he used radiometric data and he placed the Forel-Ully color scale within the chromaticity scale of our vision. So the average human would see Forel-Ully would have that color that's shown on the screen. Moreover, if you wanted to get the exact wavelength, there's this neutral white point, and you connect it through the Forel-Ully point, and it gives you the actual wavelength of color that we could use for comparing our radiometric data. So I know it's an eyeful here, but basically what Fernand did was he connected chromaticity to radiometry. So we could use that then to take all of our radiometric data to back calculate Forel-Ully. So what Henry Bigelow saw in 1912 and 1913, the Forel-Ully went between 4 and 9 here. Those were the range of colors that he saw in this book right here. And what we have seen, we go as low as Henry Bigelow did, but we go up to Forel-Ully of about 13 into the Ully scale. And the range, the average is shown about here. The range are shown by those error bars there. But what we're seeing now is a much wider range in color in the Gulf of Maine than Henry Bigelow saw in his time. And which basically says it's yellower now than it was then. And if you don't believe me, this is that tank in which we have bottles incubating in the bottom of. Remember in the very beginning I showed you the picture from the MV Nova Star where we had this white tank which had the bottles, and we keep them cold so we can bring them to the lab and work with them after we can bring the phytoplankton back. You can't even see the bottles in the bottom of that. And here's the Forel-Ully scale. And you can see, you know, the numbers. This water is collected from the very bow of the ship. There's no way they were pumping their tanks or anything like that. This is collected from the very bow of the MV Nova Star. And that's the color of the water, 6 inches of it. So you can see this is 80 kilometers from land. And it's in the middle of a water body called the Eastern Maine Coastal Current, which is carrying water from the biggest river in the Gulf of Maine, the St. Johns River. So the last thing I want to show you is the predictions for what's going to happen for DOC export over the next 80 or so years. And this is a paper that was just published by Tom Huntington and others, the guy I showed previously. And you can see, and they're using different climate models based on different emission scenarios for whether we keep pumping out CO2 at the business as usual rate or if we gradually start tapering it off. And essentially, he's predicting about a 30% increase in this DOC over the next 80 years. I didn't have time to show you. I can show you later if you'd like to see it. The slide's in here. For the past 100 years, he's also shown that it's been about a 30% increase since the days of Henry Bigelow. So, in summary, I've given you a brief introduction to Bigelow Laboratory's Charlie Yench, the founding director, and the power of satellite remote sensing for understanding the biology and the chemistry, actually, of the ocean. Didn't talk too much about chemistry, but temperatures are increasing and the hydrological cycle, the precipitation cycle, is intensifying. We're getting more floods and more droughts. I've introduced you to the 19-year time series I call NATS. There's been a Gulf-wide decrease in primary production and an increase in DOC over the last 19 years. And as was pointed out, it's not a monotonic change. There are bumps and such, which you might expect from a complex environmental system like the Gulf of Maine. Comparing NATS to radiometric estimates of color, to Bigelow's colorimetric estimates from a century before show a yellowing of the Gulf of Maine. And the prediction is that the Gulf of Maine will continue to yellow over the next 80 years. And thank you very much. Oh, I have one other thing. that I want to show you. And this goes right back to Charlie Ench's original vision of remote sensing. And there's a movie here that's booting. When Charlie died, the program manager at NASA went looking through the annual reports. You know, we sometimes wonder if the people in Washington read our annual reports, you know, and I've come to the conclusion sometimes that you know, that no one's looking at them. Well, Charlie wrote reports like people were going to read them. And so this now is a video courtesy of NASA Goddard. And we'll just leave this globe, if I can just get the movie to start. There we are, right there. And this is now scaling up from the little Gulf of Maine, right up there, to the globe. So you can see now the power of remote sensing for understanding the global ocean. And you can see major ocean currents here. And you can see, you're also looking at the color of the land as you go through seasons. We're going through entire years here. You can look at the Aleutians here in the Bering Sea, and you can see all sorts of changes going on. And while that's spinning, I'm just going to read you this little quote from Charlie. And he said, and I quote, "Without global ocean color satellite data, humanity loses its capacity to take Earth's pulse." And whoa. Oh, someone's mic on? Okay, just me. Can you still hear me? Yeah. Without global ocean color satellite data, humanity loses its capacity to take Earth's pulse and to explore its unseen world. The fragility of the living Earth and its oceans has been noted by astronaut Sally Ride, who speaks of the changes in land, oceans, and atmosphere systems as witnessed from space, and the challenge that a changing climate presents to our beloved Earth. It is our duty to provide a long-term surveillance system for the Earth, not only to understand and monitor the Earth's changing climate, but to enable the next generations of students to make new discoveries in our ocean gardens, as well as explore similar features on other planets. And that was written sometime in the mid-2000s. And you can see these fantastic changes that we can see using ocean color remote sensing at enormous basin scales, not just the Gulf of Maine, but the entire global ocean. And you'll see the Amazon and the Orinoco rivers here are putting out dissolved organic carbon, and there's chlorophyll associated with the edges of the filaments. Here's the upwelling off of Mauritania, this standard oceanography of upwelling systems where water from down deep comes to the surface and the phytoplankton are having a party. And you can look here in the northern hemisphere, what's happening right off our coast in the Gulf of Maine and in the broader scale of the North Atlantic. So, you know, ocean color remote sensing is incredibly important for what we do here at Bigelow, as well as our view of the global Earth and how it's changing. So I'll stop right there.
We'll start in the back and just wait for me.
Barney, thank you. Is there a connection between the color data that you're seeing and the temperatures of the ocean in the Gulf of Maine, and also the acidity, which we're hearing a lot about, increased acidification? Is there some connection between those data?
So we do measure the ocean acidification parameters, basically alkalinity and dissolved inorganic carbon, so that we can reconstruct, within gnats at least, the complete state of the ocean acidification in the Gulf of Maine. We were funded to start doing that in 2011. And so our database for ocean acidification is not as large. Are there connections between temperature, ocean acidification, and color? The temperature changes probably are the easiest thing to show a relationship with. And as water warms and becomes more stratified, it basically becomes less dense, and it makes for a surface layer which is harder for deep nutrients to punch into. So think about in the Gulf of Maine in the winter, it's mixed pretty much it can be mixed top to bottom. And that's water very deep, that's very rich in nutrients, that comes up to the surface, and then the next summer comes along and it stratifies that water. The plants are trapped in that upper layer with lots of light and lots of nutrients, and they say, we're going to party. And that's what they do. That's the spring bloom. Now, if you warm, in the background, if you warm that water more, so in the winter now it's warmer than it used to be, then it's harder for those nutrients to punch up to the surface, and so you would expect to see less productivity of the phytoplankton due to that warming effect. The other part with acidification, we see times during the winter months when the water is coldest and it's most soluble for CO2. The solubility of CO2 is very much related to temperature. So when in January, when the water is 4 degrees Celsius, lots of CO2 is diffusing in from the atmosphere into the ocean. And the water becomes very acid. And it's gotten to a point now in the winter months where we see acidities that are low enough, high enough acidities, low enough pH, that an organism like a pteropod, one of these shellless, it's a small mollusk, it's primary food for salmon, for example, they'll have a hard time making a shell, and the shell is their protection. So I can't, we haven't been measuring acidification long enough, I feel, yet, not like the 19 years that we've been looking at phytoplankton total, but I think there are probably, pretty soon we'll be able to see better trends, but it's a little disconcerting that the pH gets as low as it gets in the winter. We didn't expect that, and that's right across the Gulf of Maine. I mean, yeah. Oh.
Thank you. So connecting the decrease in primary productivity to the color change, is it because the spectra of light necessary for photosynthesis is not available for the phytoplankton because of the more yellowing as opposed to blue? I'm just kind of in my...
Dissolved organic carbon, the colored dissolved organic carbon absorbs heavily in the blue. And the action spectrum, not to get too technical here, but the action spectrum for photosynthesis requires blue light at about 440 nanometers. That's the peak for chlorophyll absorption. So if you bring an umbrella that absorbs all that blue light before the phytoplankton can, then in effect you would be limiting them by light. Now we will start next year actually looking at the process of light limitation by using a device which I don't want to get to it, but basically we're going to look at how limited the phytoplankton are by light, and we're also going to look at how they respond to an addition of nutrients. So every time we cross the Gulf of Maine, we're going to pick up a large volume of water, and then we're going to add some nitrate to it and look at how fast, whether those phytoplankton grow faster, which would tell us whether they're limited by nitrate. We will also take that phytoplankton sample and we'll expose it to a gradient of light intensity to look at their response to light. So we're trying to tease apart now. We're turning gnats into an environmental observatory by we'll be bringing water back every trip to do these other analyses.
What about dissolved oxygen as related to the color change?
I'm going to turn it off so you can turn it on.
The solubility of not just CO2, but oxygen changes as a function of temperature. So chances are any temperature changes in the Gulf of Maine, and I didn't mention this, but I'm happy. I'm actually going to show you another slide. Hold on. Oh yeah, here's. right there. This is a slide from a paper by a guy named Andy Pershing at the Gulf of Maine Research Institute, a fine colleague and a very smart oceanographer. And he was plotting temperature changes in the Gulf of Maine over the last 30 years in a paper that came out just a couple of years ago. And he basically showed this trend as this dashed line, and the change in temperature that he saw was 0.03 degrees Celsius per year. And you say, wait a minute, but the air is only changing 0.01 degrees Celsius per year. How could the ocean be changing faster? And these are data based on satellite only. So the satellite measurements of temperature only look at the very surface skin. And here it is. So then he looked at the change over the last decade, and the map down below, down here, shows globally the change in temperature over the entire Earth's ocean to show that, and this is over the last decade, to show that at least, even if this is not part of the long-term cycle, but this decadal change here shows the Gulf of Maine to be the fastest warming water body on Earth. So to get back to your question about oxygen, solubility of oxygen goes up as the water temperature decreases. So as the water temperature warms, the ocean will be less soluble to oxygen. There will be less of it. And in fact, there are not just trends from the Gulf of Maine. There are people that are measuring the global trend in oxygen is equally important to the global change in CO2. I don't know if that answered your question, but that's an amazing plot. And you know, our little old Gulf of Maine right there is sitting in one of the fastest warming places on Earth. Oh, sorry.
Anyone else? Oh, right here.
So are you guys measuring the water in rivers, the color of the water in the rivers, like the Kennebec? Because when I was in Bath, I noticed that the water was a really, really dark brown, almost a red.
Yeah, great question. It's all we can do. We feel sort of like one-man bands out there. We probably measure 150 different parameters when we cross the Gulf of Maine. So we know we have not extended into rivers, but people like Tom Huntington, he is a hydrologist with the USGS, and he does. And so he puts instruments up into these rivers to look at changes in the river color and changes in the inherent optical properties of that water. So you're right on the money. I mean, it's the rivers are distinctly changing. We just happen to be looking at it at the bottom of the rivers out in the Gulf of Maine, where it all goes. To that, yeah.
Anyone else? Pressing questions? Okay, well, if you could join me in thanking Barney again.